So we're going to start this unit on cells here focused on subcellular components, or from your ninth grade classes, better known as organelles. So we're going to take a look first at prokaryote and eukaryote, just a quick reminder of that, and then we're going to run through a handful of organelles. We're going to need to, you guys are going to be responsible for structure, function, how those two things tie together like we've been doing so far. Okay, so quick reminder on a prokaryote versus a eukaryote, because if we're going to be talking about membrane-bound organelles here, we need to remember what kind of cells have those. So if you remember our prokaryotic cells, these are our cells that do not have any membrane-bound organelles. Okay? These are our bacterial cells. You can see an example of one over here on the right here. There's a prokaryotic cell. They still carry out the same basic functions as eukaryotes. These cells still are able to do energy conversions. They still are able to do protein synthesis. They are still able to do reproduction. They just don't use these membrane-bound organelles to do those functions. So again, keeping in mind that these things that help sustain life, the ability to convert energy and to produce energy, the ability to reproduce, making proteins, all of those are still done by prokaryotic cells, even though they do not have these membrane-bound organelles. They do have some things, however, in common with a eukaryotic cell. If you'll remember, they have a cell membrane. They've got to maintain that barrier between inside and outside. You can see it as the plasma membrane here on this diagram. Lots of them also have a cell wall. They don't all, but lots of them do. Okay? All of your eukaryotic cells definitely do not have a cell wall, right? Animal cells don't have a cell wall. Okay? They're going to have ribosomes. Remember, ribosomes make proteins. So they've got to have ribosomes. They're going to have DNA. They're going to have that genetic material, but it won't be enclosed in a nucleus. You'll see here there's the nucleoid region where the DNA is more concentrated, but there's no membrane enclosing the DNA. Okay? Remember, in eukaryotic cells, the DNA will be enclosed within that nuclear membrane, where in prokaryotic cells it's not. They're also going to contain cytoplasm or cytosol. Okay? And all of these things are going to help contribute to these functions that keep these prokaryotic cells alive. Okay? We're going to focus, though, on eukaryotic cells. We're going to be looking at some particular membrane-bound organelles, okay, and how the way that they're built can affect their function, okay, and how these organelles working together can help make the living cell. So we're going to start first with ribosomes. Ribosomes are made of proteins and rRNA. rRNA actually stands for ribosomal RNA. Okay, so the ribosome consists of, you can see the ribosome over down here on the bottom here. Okay, the red and the kind of whitish part, all of that is the ribosome. So that's going to consist of proteins and rRNA bound together into these two subunits. Ribosomes are important for protein synthesis, in particular the translation part of protein synthesis. You can see right here how the polypeptide chain is being elongated, because the ribosome is help adding amino acids. It's using the messenger RNA. Remember, it uses the messenger RNA to help actually produce the final polypeptide chain. So it's helping it with translation part of protein synthesis. Ribosomes are also present in all forms of life. If you'll remember just a minute ago, we mentioned that ribosomes were in prokaryotic cells as well, because they are not membrane bound. So they do exist in prokaryotic cells. They do help in translation and help make proteins in prokaryotic cells, just like they do in eukaryotic cells. The significance of this is that it shows a link for common ancestry. Okay? We're not going to talk about evolution until it's going to be a few units now before we ever get to evolution, but we will be mentioning it as we move throughout. Okay? And so this is a time where seeing these ribosomes in both prokaryotes and eukaryotes is giving support to common ancestry, okay? And support to our evolution concepts. Okay? These are going to be concentrated. You're going to see a higher concentration of these in cells that are producing a lot of proteins. So that's something that you'll need to keep in mind. We're not looking here to just memorize functions of organelles. Okay? What we're looking here is to be able to apply that information. For instance, if I know that ribosomes make proteins and I know that this cell makes a protein hormone, then I should be able to make the jump that there's going to be a lot of ribosomes in that cell. So if I looked at a picture and had to choose which cell it was, I would be looking for the cell that had the most ribosomes. Okay? That's where we need to get. We need to make those jumps, make those connections between these various concepts. Okay, so these ribosomes can be either what's called free or bound. If they're free ribosomes, they're going to just be floating in the cytosol. You can see some little scattering of free ones right there, just little dots within the cytoplasm there. Free ribosomes usually make proteins that function in the cytosol. So basically proteins that are not going to leave the cell. They're going to work in the cell. They're going to be enzymes that help the cell do its regular functions. Okay? These are going to be what kind of proteins the free ribosomes will make. The bound ribosomes, you can see them labeled here, and they're stuck on this endoplasmic reticulum. The bound ribosomes, these are going to make proteins that are going to leave the cell. These are going to make proteins that are used, that are secreted from the cell, so proteins for secretion. Also proteins that are going to end up in the plasma membrane, okay, on the outskirts of the cell. But our bound ribosomes are the ones that are going to do that. So let's look a little bit closer then at this endoplasmic reticulum and why it has these ribosomes attached to it. So the endoplasmic reticulum is a big network of tubes. Okay, and so throughout this network of tubes, you'll have parts of it that will have ribosomes attached to it, and parts that will not. Okay, hopefully y'all remember that we have rough endoplasmic reticulum as well as smooth endoplasmic reticulum. The endoplasmic reticulum in general is going to help provide mechanical support, and it's going to help transport materials throughout the cell. If you look at your diagram over here, you will notice this is our nucleus, the purple in the middle here. You'll notice that the endoplasmic reticulum is attached to the nucleus and that it's a network of tubes. Okay? And so because it's a network of tubes, it allows compartmentalization throughout the cell, and it's going to be able to transport things throughout the cell. So looking at the rough endoplasmic reticulum, that's going to be the portion here that has the ribosomes actually attached to it. We mentioned before that these bound ribosomes are the ones that are making proteins that are going to be secreted out of the cell. Also making proteins that are going to go into the cell membrane. Remember, our cell membrane is mostly phospholipids with some proteins mixed in it, a little bit of cholesterol too. So this rough endoplasmic reticulum with these ribosomes attached to it is making these secretory proteins. And what it's helping do here, okay, is as these ribosomes make the proteins, these proteins are threaded into the lumen of the ER. Okay, the lumen is the opening or the space. So basically, as the ribosome makes the protein is being forced into the hole, into the tube of the endoplasmic reticulum. And once it gets into that endoplasmic reticulum, the endoplasmic reticulum can help finish folding the protein. If you think back to proteins, remember their structure was very important, how those side chains interact. So the endoplasmic reticulum helps finish folding the protein, as well as for a lot of these secretory proteins, it attaches a carbohydrate to the protein, and the protein becomes what we call a glycoprotein. Okay. Glyco, sugar, right? And so these modifications, this finishing of the folding and the chemical modification, the adding of the carbohydrate, a lot of that is happening there in that endoplasmic reticulum. And then those proteins are going to be packaged and shipped to the Golgi complex. So the Golgi complex can finish getting them ready for secretion out of the cell. It's also keeping those proteins separate from the cytosol proteins. Remember, surrounding this, you're going to have all of these free ribosomes that are making their own proteins that are going out, that are being used within the cell. And so by sending these secretory proteins into the lumen of the ER, that helps keep them separate from the cytosol ones. So again, another example of the way it's built helps dictate and helps do its function. The smooth endoplasmic reticulum, again a network of tubes, but this time there's no ribosomes attached to it. Okay? And the smooth endoplasmic reticulum is important for lipid synthesis, so making lipids, in particular making steroid hormones, oils, phospholipids. All of those are going to be made in this smooth endoplasmic reticulum. So we've got steroid hormones, which are like estrogen, testosterone hormones, oils, and phospholipids. And remember, phospholipids are what are making up the bulk of our cell membrane. So our smooth endoplasmic reticulum is helping make all of those. The smooth endoplasmic reticulum will also help with detoxification. Okay, it helps make molecules more soluble so they can be broken down easier. So you'll find, for instance, in liver cells, there'll be a higher concentration of smooth endoplasmic reticulum because they're doing a lot more detoxification. Okay, I know it seems random, but I just want to give you another brief reminder because I didn't at the beginning. Make sure that the things that are said are also getting written down. If you need to draw pictures, draw pictures. Remember, these are your notes. Okay, if you're only writing down what I have typed on this slide, that won't cut it. You won't get there. Okay, so now let's talk about the Golgi complex. So these proteins were produced in the rough endoplasmic reticulum. Okay, and then they're going to get shipped over to the Golgi complex. The Golgi complex is going to help make sure that these proteins were folded correctly, that they have the correct chemical modifications when we talked about those glycoproteins, getting the carbohydrates added to them. Okay, so we're making sure they're folded properly, they have the right chemical modifications, and that they're packaged appropriately. They're trying to ship these out of the cell. These common example of these proteins that are being made, that are being shipped out, are hormones. They're going to travel a long distance in a multicellular organism. So they need to make sure that they're packaged properly so that they can be shipped out. This Golgi complex has a distinct polarity. If you remember when we talked about DNA and when we talked about proteins, we talked about their terminality and them having a directionality, that they had a certain end where only, like for instance, the 3' end in DNA is the only end you can add nucleotides to. Okay, so this Golgi complex is also built in a similar manner. It has a distinct polarity. And so basically what this does, and we're not going to get into a lot of detail here, but the way these membranes are laid out is it forces vesicles to only enter on one side and to exit on the other side, so that they'll move through the complex, getting there, making sure their folding is correct and they have the right modifications and the right packaging. And it basically creates a one-way street for them. Okay, this polarity. So again, another example of the structure and the function going hand in hand. By creating this one-way street, okay, it ensures that they're moving properly through the Golgi complex and getting checked and sorted out. Okay, and there's not like a log jam of proteins in there. Okay, so now that we have these proteins built and exited out of the cell, let's take a look at vacuoles. Vacuoles are another membrane-bound organelle. These are just membrane-bound sacs. They have a wide variety of functions. Okay, they can act for food storage. Okay, we've got certain cells and certain organisms that are only unicellular. They take their food in by the process of phagocytosis. Okay, if y'all remember, phagocytosis was cell eating, right? Where it, for instance, if you had like an amoeba, right, and the cell would reach out and grab the food and then close around it. Okay, and so that food then would be stuck in a food storage vacuole. Okay, you have certain cells in your body, like macrophages in your immune system. That's how they're going to collect the foreign invaders. Okay, they're going to help clean up the dead material. Is they'll bring that in through phagocytosis. You also have vacuoles that will act. They are what are called a contractile vacuole. Contractile vacuoles help maintain homeostasis. They help maintain salt concentrations in, again, unicellular organisms, as a general, unicellular organisms that are living in water. And so what will happen is these contractile vacuoles, they will pump out excess water. And so as the organism is monitoring its salt and water levels, it can easily pump out the water. It can't as easily remove the salt. And so it can remove excess water to help maintain salt concentrations. Remember, these organisms are living in a water environment, okay? And so a lot of times they are in a hypotonic environment, and so water is moving into the organism, and so they need to pump out the extra water. In plants, we have a central vacuole, which is found only in plants. Again, it's not found in animal cells. That central vacuole will do a variety of things. It can help store materials for the plant. It can help store metabolic waste that maybe wouldn't be great to have it floating around in the cell. So it can help store metabolic waste. It can store inorganic materials and nutrients for the cell, and it can store water. And by storing that water, it can help maintain the turgor pressure of the plant. Remember, as the water goes into this vacuole will get bigger and bigger and bigger. It will swell, and that will push on the cell walls of the plant, which is what makes the plant, remember the plant cell, when the vacuole is pushing on the cell wall of the plant, right? Those cell walls push against each other, and the plant doesn't look wilted anymore. Okay, the plant looks nice and healthy, turgid. Okay, and so the central vacuole helps maintain that turgor pressure in the plant, and we'll talk more about that when we talk about cell transport. So in addition to vacuoles that are going to help with the storage of things, you have lysosomes, which are also just little membrane-bound sacs, but this time they have hydrolytic enzymes in them. Okay, if you look at your word here, right? Lytic, break down, burst, right? We already talked about hydrolysis, where when I add water, I break a polymer into its monomers. So these hydrolytic enzymes help break things down. Okay, basically they're like little stomachs. And so these lysosomes, inside the lysosome, the pH is different than it is in the rest of the cell. The pH is lower. Okay, again, I know a lot of y'all are just now getting into chemistry. Okay, so when our pH goes, a quick pH review real quick, right? Remember the pH scale goes from 0 to 14, with about 7 being neutral, and lower numbers are more acidic. So the closer I get to 0, the more acidic it gets. So the more dangerous, the more caustic it would get. And alkaline, sometimes called basic, gets stronger as it goes to 14, which makes sense. If 7 is neutral, right, the further I get away from neutral, the stronger the acid or the base gets. So 14 is my strongest alkaline solution, and 0 it would be my strongest acid. And so inside these lysosomes, the pH is a little bit lower. It's more acidic than the rest of the cell, because these hydrolytic enzymes, which are basically digestive enzymes, they work better in a lower pH, just like in your stomach. You have a lower pH in your stomach because the enzymes work better at that function. So these lysosomes are important for remember, within the cell. So digestion within the cell takes in a food particle. Well, that food particle is just going to sit in the vacuole unless a lysosome comes over and attaches to that vacuole and starts breaking it down. So important for intracellular digestion, important for recycling materials within the cell. Let's say you have an organelle that dies or gets damaged. The lysosome will fuse with it. They'll form a vesicle around it. Let's say you have this mitochondria right here is broken down. So a vesicle forms around it. Then the lysosome will fuse with that vesicle and break that old mitochondria down. And now your cell can reuse those parts. Okay? Lysosomes are also important for apoptosis. Remember, apoptosis was programmed cell death. Okay? And so when a cell is set to go through apoptosis, then those lysosomes can release those enzymes out into the cell, which will help with that apoptosis process. All right, so let's shift over to our two organelles that help with energy conversions. Okay, these are going to be really important, obviously, to the cell. They're doing energy conversions. Okay, and so we're going to look at a mitochondria first. So a mitochondria has a double membrane structure. Okay, so let's look at what we've got going on here. We've got an outer membrane. So an outer membrane here, which is this kind of darker looking shadow part. So we have an outer membrane, and then you have an inner membrane. And you'll see this inner membrane is all wiggly, right? It's wiggly and it has a bunch of folds in it. Those folds are called cristae. So these are the folds, like all these little wiggly folds here. Those are the folds on the inner membrane of the mitochondria. That's where you're going to find those cristae. Those folds are going to be important because those folds increase the surface area, right? If this was my mitochondria and the inner membrane was just a circle there, and I unfolded that inner membrane flat, it'd be, I don't know, that long. But if I take this inner membrane that's all wiggly and I unfold it flat, it gets way longer. So these folds are important because they're going to increase the surface area. And on that inner membrane, that is where the mitochondria is really making ATP. Okay, so on that inner membrane is where you're going to find those electron transport chains. And those electron transport chains are what are actually synthesizing the ATP. So if I have more folds there, if I have more surface area, I can have more electron transport chains. Therefore, I can do more energy conversions. I can make more ATP. So structure helping with the function again. Okay, and then inside this area is our matrix. So inside that inner membrane there is going to be the matrix area. So this mitochondria is compartmentalized. It's separated into different areas. The benefit of separating it into different areas is that it allows for different reactions to happen in different spots. Okay? For instance, in the matrix is where the Krebs cycle is happening. If y'all remember cellular respiration, remember it all started with glycolysis. Okay? And then if oxygen was present, that pyruvate moves into the mitochondria. Okay? And so then in that matrix is where the Krebs cycle is going to happen. Okay? And that Krebs cycle is going to help produce electron carriers that are going to help power those electron transport chains. Okay? You're going to find mitochondria in all eukaryotic cells. So I know we really get drilled into, plants do photosynthesis, everybody else does cellular respiration. Well, these mitochondria are doing cellular respiration, and you find them in all eukaryotic cells. A plant can make all the food in the world. It can do photosynthesis all day long and make ridiculous amounts of glucose. But if it never turns that glucose into ATP, then it is doing no good. There's no point. In addition to the membranes in the matrix and the inner membrane space, the mitochondria also contain their own DNA and ribosomes. Okay, that again is going to be important when we talk about evolution. Okay, if you remember from 9th grade year, we talked about endosymbiotic theory, the fact that these larger prokaryotes had engulfed a smaller one, and that eventually is what led to eukaryotic cells. Okay, the mitochondria having their own DNA and ribosomes help lend support to that theory. The fact that it has DNA, it has ribosomes, it makes its own membrane proteins. It doesn't make all of its proteins, but it makes its own membrane proteins using its DNA and its ribosomes. So again, lending support for common ancestry and lending support for endosymbiotic theory. So tying this evolution in as we move through. Okay. Also, don't forget, this would be a great time to make a picture. Maybe you haven't drawn any pictures yet, and that's cool. I get that. Okay. However, we now have an organelle that we pointed out parts of, that we said what the different parts did, so maybe that would be important. Okay, so just kind of keep that in mind. Pictures will help you. So let's look at our last organelle here. Let's look at a chloroplast. So a chloroplast also has a double membrane. It has a double outer membrane. You can see the double layers here. So it's got a double outer membrane. Chloroplasts are going to be found in plants and photosynthetic algae. Remember, you don't find chloroplasts in fungi. Okay? Fungi do not do photosynthesis, therefore they do not have chloroplasts. So plants and photosynthetic algae. Right there next to the mitochondria, because the mitochondria are going to be in both of those things as well. Okay, so in the chloroplast, I've got my inner and outer membranes. Okay, so inner outer membranes there. Then I also have these thylakoids, and those thylakoids are like the individual pancakes here, okay? And then a granum is a stack of the thylakoids. So I have those thylakoids with a granum. A grana is multiple stacks of pancakes. And then I have the stroma, which is the liquid that surrounds all the thylakoids and the stacks. Okay, again, hopefully this is all sounding familiar and starting to come back to you from your 9th grade year. So these thylakoid membranes are going to be important because these thylakoid membranes, these are what contain the pigments. And the pigments are what capture the light energy. Remember, the job of the chloroplast is it's an energy conversion situation, right? The chloroplast is doing photosynthesis. It's converting light energy into chemical energy. So the thylakoid will contain those pigments. And those pigments are what are going to actually capture the light energy. And so just like the mitochondria, by having these stacks in these folds, that increases my surface area. If I just had a straight stack, then I don't have as much space for these pigments. But by having these thylakoids on top of one another, I have a lot more space for these pigments to be on the membrane. So I can do more photosynthesis and do more of these energy conversions. The stroma is where the carbon fixation part takes place. So this is where you're actually forming the sugars. Okay, so if y'all remember, again, from 9th grade year, we talked about light-dependent reactions. Those are happening on the thylakoid with the pigments versus the Calvin cycle happening in the stroma. So where the actual formation of the organic compounds happens. Okay, and by having these different separate compartments, having all these different membranes in there, it allows for these different functions. It allows for one area to be capturing the light, one area to be producing the carbon compounds. Just like mitochondria, chloroplasts also contain their own DNA and ribosomes. So again, showing that evolutionary link, that support for that endosymbiotic theory. Okay, remember, endosymbiotic theory. Okay, so these are the organelles that we're going to focus on in the eukaryotes. Make sure you understand their functions. Keep in mind, think about how they could work together. Don't treat them as completely, totally separate things. Okay, if you needed to draw pictures, go back and get some pictures. Okay, and we'll be working with them in class.